Many thanks, Roy. It's my pleasure, really, my pleasure to be here. And really, I like to just focus what I'm going to say to you based on probably the following fact that's probably true, that of this entire room, I've conducted more unsuccessful clinical trials than anyone. Having been my entire career a phase 1 doc with hundreds of publications that could easily have never been published and never have affected the course of cancer therapy. And I'm going to do so from a historical perspective. And because over the last year and a half, we've been engaged with a group of academic partners and industry partners that really focuses on following hopefully this is true too, historical perspective. And that is, if you look back over the last 100 years of cancer research and the models used for cancer research, I think it breaks down in a very simplistic sense into 3 eras. You start at the turn of the 20th century, 1900s, and basically our models were patients. We learned how to do mastectomies and give radiation therapy with patients. We actually learned how to give the initial cancer chemotherapeutic agents not from any model system other than patients. And then the availability in the mid-50s, late 50s of quote-unquote reproducible murine models of leukemia allowed us to switch to a model-focused approach. And you can't say that it wasn't a useful exercise, because combination chemotherapy for lymphomas and many leukemias derived from the kinetic approaches that were studied extensively from the late 50s through, let's say, the early 80s. And then those model-focused approaches hit a wall when, after discovering essentially every potential cytotoxin that was useful in those models, we tried harder and harder on larger and larger panels of plastic-based cell lines, and we found some important biological leads. But that has also led, I think, easily in the last 10 years, probably more in the last 5 years, to the notion that, at least I would like to put forward the notion that what we need are individualized patient models. That we need models that really, much more specifically, but to the best of our ability, they won't be perfect, actually look at what's going on in an individual patient and help us derive approaches to treatment in individual patients. Whether those are PDX models, circulating tumor cell models, biomarker-derived models from biopsies of patients, or all of the above, that's what I'm briefly going to talk about. And I think that this consortium that we've been trying to work with and bring together to put together an entire repository of models that the NCI could distribute to investigators around the country and around the world, whether they are Rho kinase-derived models, PDX models, CTC models, is something that I hope we will find out. And I think, as was discussed at the end of the last panel, the issue is, do I know that these things are going to work? I certainly don't. But do we need a way to find out how to do better than the previous xenotransplants that my division in particular has used for 25 years and are clearly unsatisfactory? So let me just give you some examples of what I'm talking about. So this is a patient, actually a patient treated in my clinic with advanced colorectal cancer, had 2 biopsies, May 1st, May 22nd, grew out PDXs, same histology as the original tumor. More importantly, a single blood draw using a non-degrading technique to develop and separate colon CTCs also grew out an identical PDX. As importantly, one can get from the biopsy, this is a single biopsy in an 18-gauge needle biopsy, or from blood, also Rho kinase cell lines that can, as you'll see on the next slide, be put back into a mouse and generate a xenograft from a Rho kinase cell line that's identical to the initial xenograft PDX and the initial patient. And then, of course, when you have these kinds of materials, they're not simply 1 or 2 or 3 passes of an 18-gauge needle. One can then do a variety of different kinds of biology, I would hope. There we go. Whether you do whole exome sequencing or do a series of validated panels from the original biopsy, from the PDX, from multiple generations of PDXs, from circulating tumor cells, from the solid tumors generated from those cells, or from row kinase cell lines, to tell you something about the heterogeneity and to help you characterize those, if you will, mouse patients. I was telling someone that I'm really glad I had a lab meeting on Friday, because this actually comes from data from Friday in which this is a bladder cancer with a well-characterized P53 mutation that is the binding site for P53 from a PDX we developed from a patient. And it shows, and basically this, as you'll see, is a prelude to a large clinical trial that we're conducting, but we're conducting it in a series of mouse patients. Unlike the clinical trial, we can give a series of these mice all of the various arms of the trial. And we show very clearly that the DNA damaging agents produce significant regressions, and I don't know if this will be a cure or not, but the MEK inhibitor doesn't do anything to this well-characterized tumor. And so I think that we are on the verge, if not there already, of being able to go from a CTC to a mouse model to a PDX to a therapeutic model, and back and forth when we do these kinds of things. And so this is a trial. This is not the MATCH trial. That's a bigger study that the NCI is conducting, but it is what we call our M-PACT trial, which is an umbrella for a randomized prospective evaluation of 4 different types of therapy where patients get an initial biopsy. They get a biopsy at progression, and they're randomized to get either a matched treatment initially or a matched treatment at progression. And so we will have, at the time these patients are initiated, a mouse model that will develop from the patient. We'll have a mouse model that will produce from a biopsy at disease progression, and we will hopefully be able to utilize, beginning early next year, this process of going back and forth to understand whether these new models that really do things that are not the same as xenografts, whether patient-derived materials can be the focus for this back and forth so that we understand we're no more than a few months behind the patient in devising what kind of treatment to engage in. And so, so my 3 points really are these. And it's not just that whether should we go ahead and do co-clinical trials. I mean, that's going on. But the question is, can we develop essentially for every patient? At least initially, I'm optimistic that at least half of the patients will be able to develop not just a population base to go after of mouse tumors that come from patients, but individual preclinical predictive information that we can translate. Can we take that from all the various models that we develop in a comprehensive fashion and utilize those in clinical trials? And can we develop the collaborations necessary across all of the entities that we've talked about earlier, the earlier panel, to make these kinds of things on large scale available, both for individual patients, but also to provide the data that we need to model these very complicated systems.
Great, thank you very much. We have time for perhaps one question. If you could go to the microphone, because there are a large number of people listening online to this meeting, please.
So Jim, obviously we embrace much of the same vision here, but the question is whether, given that one would expect not to be an infinite number of mechanisms for the etiology of a particular tumor, and therefore for chemosensitivity, that you don't necessarily in the future will have to actually grow a PDX for every patient. So are you thinking about creating sort of a data bank of PDXs that then new patient can be matched against?
So our goal is to develop somewhere between 1,000 and 1,500 PDXs, clinically annotated, fully characterized, that are in the public domain, that will have not only all of their molecular information, but also a lot of therapeutic information that we can then use as, if you will, templates.
Bill?
I think unfortunately, as you know, the history of clinical oncology is littered with things that we were absolutely certain would be helpful, and we were so certain we were reluctant to even do the proper clinical experiment, but lo and behold, when we did the proper clinical experiment, we got a disappointing result. So I wonder if once and for all, for example, the clinical trial you showed, the people who had the mutation are randomized to targeted therapy versus, I guess, standard of care, but the people who didn't have the mutation didn't enter into the randomization.
Correct.
That sort of presumes that the genetic alteration is a potentially a predictive biomarker. I mean, isn't the proper experiment to likewise randomize the people who didn't have the genetic marker to standard of care versus targeted therapy to once and for all know whether or not, for example, there isn't a significant response rate amongst those people who don't carry the mutation?
So the way that this is basically, it's not across standard of care. So there are basically 4 or 5 strata of targeted therapies. You either get the matched treatment or you get something that would be predicted not to be useful initially. And at progression, you cross over. And actually, Bill, the trial is powered so that all of those strata, when added up, will help us understand whether matching therapy or not initially changes your PFS.
Yeah. Well, I think my general comment still holds, if that's the design. Then you're really relying on historical controls in part. At least that's the way some of the bucket designs look to me. But maybe we can discuss this offline.
Sure. Right, and we'll probably discuss more of that on the panel once we get done. Well, thank you very much, Jim. So our next speaker is from the FDA, and we'll change the order a bit, and it will be Dr. Elizabeth Mansfield. And Dr. Mansfield is the Director of Personalized Medicine staff in the Office of In Vitro Diagnostic Devices and Radiological Health in the Center for Devices at the FDA. And she's going to talk to us a bit about her perspective on this topic.